Okay, hey microclass. So today we're going to continue on in chapter 12 with adaptive immunity. Okay, so remember last time we covered innate immunity, and that is something that you are born with, something that happens immediate, and it's not specific for a particular type of pathogen. Okay, it's just going to keep everything foreign that's identified as out. Okay, so it's not going to look for specific pathogens. Okay, also the innate immune system includes the first and second lines of defense. First line of defense is the physical barriers like the skin and mucous membranes, and the second line of defense includes different types of cells like basophils, eosinophils, neutrophils, phagocytes, and then also some chemicals and processes like fever and inflammation. So chapter 11 looked at our first and second line of defense. And today we're going to get into the third line of defense, the adaptive immune system, which includes cellular responses, which is going to be T cells or T lymphocytes, and B cells or B lymphocytes. Remember, the term leukocyte is basically a fancy term for white blood cell. Lymphocyte, like T or B lymphocytes, are types of leukocytes. So try to keep those terms separate, and then also definitely know the differences between adaptive and innate immunity and what is involved with each of the three lines of defense. And they all three basically work to keep the body safe from invaders. Okay, so the adaptive immune system responses closely interact with innate immune responses. You know, they work together to keep us safe. And if any part of either the innate or the adaptive immune system is impaired, if it's not working properly, that person is considered immuno or immune, either one, compromised. Okay, and that person would be at risk of infection, at a greater risk of infection. Okay, so adaptive immune responses go into effect when innate first and second line defenses fail. Okay, so remember we said the first line of defense is kind of like a barrier, like a barbed wire, razor wire fence around the fort. Okay, that's the skin, that's the first line of defense. If a pathogen breaches that first line of defense, fever and inflammation will try to help. And if that's not working, if phagocytes are not working, the third line of defense, the adaptive immune system, will also begin working. Okay, now adaptive immunity, remember, has memory. Okay, it remembers a pathogen. And so it's going to be different from the innate responses because it's going to remember that pathogen later. Okay, so when we talk about the adaptive immune responses, we talk about primary response or primary exposure and then secondary exposures. Okay, now adaptive immunity takes longer for it to come into effect. Remember, innate immunity comes really quickly, pretty much immediately. This adaptive immunity takes a few days to get into effect. Okay, so the first time, the very first exposure a patient has to a particular pathogen, it takes a week or more, maybe closer to 2, to really get a good response from the adaptive immune system. Okay, and so one of the differences is this adaptive immune response takes longer to mount or to get going. Okay, also remember, it is going to be looking for a specific antigen. And remember, we said an antigen is any substance that elicits or turns on or stimulates an immune response. Okay, So after the first exposure, the body is going to remember that antigen associated with that pathogen. Okay, so a major advantage of the third line or adaptive immunity is going to be that memory. Okay, so secondary exposure, or basically 2 or more exposures to the same antigen, is going to cause a rapid and more effective response by the immune system. Okay, this is why we give vaccinations. Okay, and we'll talk about, we have a chapter coming up, chapter 14 gets more in depth into vaccinations. But vaccines are developed to give the body that initial exposure without the chance of getting sick and getting the symptoms. So it exposes the body to antigens so that it can make memory. And then if you're exposed to whatever you were vaccinated for, say you were vaccinated for tetanus, and you were exposed to tetanus by stepping on a rusty nail, then your body's like, oh, I know what that is. I'm not, I'm going to gear up and I'm going to take it out right away. And then a lot of times you won't experience symptoms, that you'll have the pathogen in your body, but the pathogen doesn't have a chance against your immune system. Or if you do experience symptoms, they are going to be quite a bit weaker than if you hadn't had that initial exposure. Okay, now, as I mentioned, the adaptive immune system is divided into 2 categories. Okay, the first one is called cellular immune response, and that's going to be the T cells that are going to be working, the T lymphocytes. Okay, sometimes that's also called T cell mediated immunity. Or there's another branch called the humoral response, or antibody mediated immunity. And this is going to be the B cells that produce antibodies. Okay, they both have the same goal, is to get rid of that foreign substance. Okay, they just go about it in different ways. Okay, now they both go through 4 main steps to get there. They're just a little bit different in how they respond. Okay, so this particular slide is on page 352. And if you want to pause the video and grab your textbook, if you have a hard copy of the book, that might be beneficial as we're kind of going through some of these slides, because I know on your handouts are really small. So you might want to grab your book and just look through this. And I would really encourage you to stop this video as we go through and think through the processes, think through the stages, think through the different types of cells, and help yourself make it make sense before you move on. Okay, so this might be a place where you stop and go back and think about the different aspects of adaptive immunity and how it's different than innate immunity. And then when you're ready, you can move on, and we'll talk about these 4 stages that both the B cells and the T cells go through to protect the body with adaptive immunity. Okay? All right, so in order for T cells and B cells to protect the body, they have to first identify what the antigen is, identify what the threat is. And so that's going to be stage 1. Okay, they are going to find an antigen that's been presented by a phagocyte. Then they're going to be activated by chemical messages. Then the T and B cells are going to reproduce. They're going to make a bunch of clones of each other. And then those cells are going to differentiate. Some are going to become memory cells, memory T or memory B cells, and others are going to be what we call effector cells, or the actual soldier cells that are going to go out and eliminate the pathogen. And then the 4th stage is basically when the pathogen elimination begins or takes place. Okay, so we'll walk through those steps here fairly briefly. So stage 1, okay? Dendritic cells, which are phagocytes. Remember, a phagocyte is a white blood cell that's going to go around and engulf foreign invaders and digest them. We talked about those last time. If you need to, go ahead and Google or search for a YouTube video on phagocytes. Okay, phagocytes go, they engulf the bacteria that's foreign and destroy it. Okay, these phagocytes, like dendritic cells, are what we call antigen presenting cells. Okay, and these cells show the antigen to T cells and sometimes B cells. Okay, so if we move back up here, if we enlarge this, and dendritic cells have kind of a crazy shape to them like this, and they have ingested a bacterium. Here's maybe E. coli, and on this E. coli they have a round little antigen, this little structure right here. As this phagocyte, the dendritic cell, and this is in a vacuole, digests and breaks down this E. coli, it's going to take this antigen and display it out here. It's going to present, oops, the antigen. I'm going to abbreviate it AG because this pen is terrible. Okay, it's going to present this antigen out here, just like we see up here. Okay, it's basically waving the red flag like, hey, I found E. coli right here. This guy is foreign. Here's his antigen. Somebody come and help clear out this bacteria, because where there's one, there's going to be more. Okay, so the antigen presenting cell, which is a phagocyte, part of the innate immunity, see how they're working together? Innate immunity antigen presenting cell is going to come over here, and then this T cell is going to come over here, say, oh, I recognize this guy. It becomes activated through chemical messages and the process continues. Okay, so take a second and stop and review antigen presentation if it doesn't quite make sense. Step 2, okay, once that antigen is presented, the lymphocytes, which again are the T and B cells, are going to become activated by cytokines. Do you remember that term from last time? They're chemical messages. Wow, really terrible to write on the screen. Okay, now special T cells are going to also influence or activate B cells. And we'll see how that works when we get into the B cells. Okay, stage 3, that is when those cells are going to go under multiple rounds of cell division. Okay, they're going to undergo mitosis, and this process is called clonal expansion. They're going to basically make a bunch of clones of themselves. Okay, because each of these T cells and B cells can only identify one type of antigen. Okay, so if there is an E. coli infection, we want to make sure we have a lot of T and B cells that can fight this particular antigen-bearing E. coli. So once the T cells that have the right recognition system are identified or activated, we need to make more of them. We don't need to make just T cells in general. We need to make more specific T cells. And so that's what this clonal expansion does. It takes the ones that know how to fight this particular pathogen and makes more of just those. Okay, so when this clonal expansion occurs, when the cell division is occurring and all of these new T or B cells are being produced, majority of them are what we call effector cells. And these are the ones that are going to engage. These are the ones that are going to kill or take out the antigen-bearing cells. Okay, so most of these new clones are going to be effector cells or warrior cells, and then a few of them are going to be memory cells, so that way if you step on that rusty nail later or you get exposed to this E. coli again another time, they're going to remember this antigen. They're going to remember this response and help boost the body's response to that antigen. Okay, and then step 4, that's actually like the quote unquote killing period. Okay, it's going to be eliminating those threats. Okay, so the T cells and the B cells are going to be responding during this time, and we'll talk about how that happens. And then once the threat passes, the effector cells begin to die off. Okay, the killer cells are going to start to die off. And then the memory cells might stick around for years. Okay, months or years, just depends on a lot of different things, but could be months, could be years. Okay, oops, I wanted to come up here too. Back up one slide. Effector cells are going to be what we call cytotoxic T cells, or sometimes called killer T cells, and then also what we call plasma B cells. Okay, those are the two main types of effector cells in the adaptive immunity: cytotoxic T and plasma B cells. Okay, so we've talked about the lymphocytes, the T and the B cells. And remember, leukocytes are white blood cells. Lymphocytes are these T and B cells. Okay, some differences between T and B cells. Both T and B cells and most blood cells, red blood cells, most white blood cells, excuse me, are produced in the red bone marrow of the long bones. The T cells are going to mature in the thymus. The B cells are going to mature right there in the bone marrow. Okay. Mature T and B cells are pretty low in blood circulation. There's not a lot of them that circulate around in the blood, a few, but very little. Most of them are going to remain in the lymph nodes and other lymphatic tissue. Okay, the lymphatic system is going to be basically the stage for where most of the adaptive immune response is going to happen. And so they're hanging out in those lymph nodes and other lymphatic tissues to figure out what is going on and to do surveillance. Okay, so T cells, especially what we call helper T cells, have roles in both humoral and cellular immunities. Okay, B cells are going to basically work to make antibodies. Okay, the antibodies is actually what's going to do the work at eliminating pathogens when it comes to the humoral immune system. And we're going to talk more about antibodies toward the end here. Okay, so an antigen, we defined that last time. Remember, that's any substance that elicits, or let's use this other word here, triggers, starts, causes an immune response. Okay, could be proteins, could be sugars found on bacterial flagellas, their cell wall, could be a toxin that they produce, or proteins and sugars associated with viruses, fungi, protists, or anything else like that. Also, cancer cells a lot of times will produce antigens as well that the immune system can identify. Okay, I've skipped quite a few cells because we're doing this remotely, or slides, because we're doing this remotely, and I want to keep it as simple as possible because, trust me, I hate teaching the immune system this way. Feel like the last chapter was not my best work, so sorry about that. But we're going to skip some stuff to just try to keep this as basic and simple as possible to give you the information that you need to know, but not to overwhelm you with a lot of crazy details. Okay, so we're going to skip ahead to this slide on epitopes. Okay, so we have an antigen, right? Here's our E. coli, and it's got this little antigen on it. If we zoom in on the antigen, we have what's called an epitope, and that's going to be a three-dimensional part of an antigen that is going to be recognized by a T or a B cell. Okay, so the antigens basically hold, so this whole thing here is an antigen from HPV, human papillomavirus. Okay, it's kind of like a model that they've made. On this antigen, you can see several different colors. There's several different epitopes or several different regions here that can be recognized by a T or a B cell. Okay, so that's actually good for the body because you may have a T cell that can recognize this epitope, and another T cell recognizes that epitope, and they're both undergoing clonal expansion. They are looking at different epitopes, but it's the same antigen, and you're getting more of the response. Maybe you have another one here that a T cell is working on, and maybe you have a B cell that's producing antibodies for this one. And so this is basically just a piece of the antigen. Okay, so the antigen's a piece of the pathogen. The epitope is a piece of the antigen. Now on the T cells and the B cells, we have drawings of them here and down here. On those cells, they're going to have thousands of these receptors. Here's a zoomed in view. Okay. Each one of those receptors on a particular T cell is going to be the same exact shape. So one T cell can only identify one type of epitope. Okay, they may have 1,000 receptors here, and they could bind potentially, probably not in likelihood, but they could bind to many different epitopes. Okay, that's not right. They could bind to many different antigens that have the same epitope. So right here, maybe they could bind to one bacterial cell with this triangle epitope. Maybe they have another triangle epitope bound to the same type of bacteria over here. Does that make sense? But they have to be all bound to the same epitope. Same thing with B cells. Each B cell may have 1,000 or more of these little receptors on it, but it can only attach to one type of epitope. And the difference, there is a difference between T cells and B cells, and the B cell receptors, you've probably noticed, have two binding sites. So they could bind to an epitope here, and they can bind here. So they could bind to a bacterium here, and here, and here, and here, and really just grab on. Okay, so the T cells and the B cells have receptors that are going to identify foreign invaders. Okay, so we've said that they can only recognize the same one type of epitope though. Okay, same one specific epitope. Okay, and that's why they need to be cloned so we can make many, many copies of the correct T and or B cell. Okay, so when a T or B cell binds to a specific epitope on an antigen, they become activated, and that's done by cytokines or those chemical messages. Okay, once that binding happens, the cells release chemical messages that signal the clonal expansion or other processes to take back. Okay, and so remember, some of the clones develop into effector cells while others become memory cells. So with B cells, when B cells clone, they're going to mature and differentiate. Okay, differentiate means it's going to take on a specific role. Okay, and if a B cell differentiates into an effector cell, this B cell is then called a plasma B cell. And these plasma B cells make antibodies. That is their job, is to make antibodies that go on and then do the fighting. So the antibodies are going to be the fighters. Okay, the plasma B cells make the fighters, make the weapons. Okay, T cell activation leads to differentiation, and there's different types of T cells. Okay, there are cytotoxic T cells, and here's an abbreviation there, that these are going to directly destroy infected cells or cancer cells. Okay, so when a T cell, a cytotoxic T cell attacks, it's going to attack the host cells that have either gotten cancer or infected with a virus or an intracellular pathogen, where the pathogen is inside of the host cell. So a cytotoxic T cell will say, "Ooh, that my host's, my person's lung cell is infected. I'm going to take out that lung cell. I'm going to kill it along with the virus inside of it, and eliminate the infection that way." Because remember, when our cells are taken over by viruses, they're not doing their normal job anyway. Okay, so that's what cytotoxic T cells do, is they destroy abnormal cells or infected cells that are part of our body. Okay, now there's different types of different types of T cells, but we're going to just focus on a couple. So that's the killer T cells, or the cytotoxic. Remember, cyto means cell, toxic. Okay, killer T cells. Then we have helper T cells. These guys don't actually destroy invaders, but they're going to coordinate the adaptive immune response of the cytotoxic T cells and the B cells. They're going to be basically like the organizers of the war. Okay, and then we have what's called CD proteins. Basically, that's a way to just determine whether a cell is a helper T cell or a cytotoxic T cell. I'm not going to make you memorize any information about that. But I would note that CD4 cells are what HIV targets. So they're helper T cells that HIV targets. They don't target the B cells. They don't target basophils. They don't target cytotoxic T cells. They attack the CD4 portion of the helper T cell. Okay, so these T cells help coordinate the immune response by releasing those chemical messages called cytokines, and they're going to activate T cells and B cells. Okay, their job is to activate and coordinate this attack. Okay, so cellular immunity, cytotoxic and helper T cells. Humoral immunity is going to be the B cells. And the helper T cells are going to release chemical messages that activate B cells and that also activate killer or cytotoxic T cells. Cytotoxic T cells, again, destroy cells that are infected with a virus or other, like tuberculosis is an intracellular pathogen. Cancerous cells, or if you get a transplant from someone that wasn't a match, they will attack those as well. Okay, I'm not going to have you guys memorize this process or look into this process, but T and B cells are screened for self-tolerance. Basically, if you've ever heard of an autoimmune disease or an autoimmune disorder, what happens is the immune system doesn't recognize normal healthy cells as normal healthy cells. The immune system thinks that it's a threat, and your immune system starts attacking your normal cells. Okay, so there are autoimmune diseases where this happens, but for the most part, there's a fail-safe where T and B cells, before they're released out into the body, are screened and undergo testing, if you will, either in the bone marrow or in the thymus, to check to see if they're going to attack self. And if they're going to attack self, they get destroyed. And that's what some of these stop signs are for, showing that those cells would be destroyed if they're going to be what we call self-reactive or destroy normal healthy cells. Okay, so just with this, just know that there's a testing that goes through to try to make sure that those T and B cells are not going to be what we call auto antibodies or auto self-attacking. Okay, so one way that we can determine if a T cell is going to attack another type of cell is by looking at or screening its protein called major histocompatibility proteins. And these proteins are specialized self proteins. So they are unique to an individual. Okay, so if you have a identical twin, you'll probably have the same MHC proteins. And there's different types of MHC proteins. There's type 1, type 2. I'm not going to make you remember which one does which, but I wanted to point this out because it is these proteins that they look for when you do a tissue match or you're doing looking for a match for an organ donor. Okay, if these MHC proteins between the recipient and the donor don't match, the recipient's immune system will start destroying cytotoxic T cells, will start killing that donated organ. Okay, so this MHC complex, that is going to determine whether an organ donor is a match or not. Okay, and it's one thing that the immune system looks for to determine if something is foreign or not. Okay, so we're going to skip this one and jump into a comparison of T and B cells. So this, I don't have the page written down, but this would be a good table for you to review. So in the T cells, okay, the cellular immunity and the humoral over here. Okay, T cells matures in the thymus. We've been over a lot of this, so take some time and review it. This might be a good time to stop the video and look at the differences between T and B cells. And then we'll move on. I'm going to skip a couple of other cells as well. This talks a little bit more about MHC cells. A couple more slides. This talks a little bit more about MHC proteins. Let's see. This kind of gives us an illustration of how intracellular antigens are exposed. So here is a phagocyte, like that dendritic cell I was showing you. And here's the antigen from that, say, virus that was living inside of, oops, I'm sorry, whoa, back, I got way off. This is not a dendritic cell. This is an infected cell, an infected normal cell. Okay, sorry, I was a little bit off there. So if this cell is infected by, say, influenza, that cell will then send out a red flag, and could also present the epitope on the antigen. And the T cell will come and identify that. Okay, so this is for intracellular pathogens. Extracellular pathogens is where we have that dendritic cell, that phagocyte that's going to present the antigen here on its outside. But this process, the antigen presenting process, is basically just like waving a red flag saying, hey, I've identified this threat, come over here and help me take it out. Okay, I'm going to skip that slide. This just talks about, don't worry about allo recognition. This just talks about recipients and donor matches and all that kind of thing. So you can read through that if you're interested in tissue or organ donors. Okay, so stage 1 with T cells, antigen presentation. Stage 2 is the T cell has to be activated. Okay, so you can see the cytotoxic T cell here is attached, the helper T cell here is attached. Okay, and this is on page 360. We're on stage 2, page 360, if you're following along in your book. And due to TCR, or T cell receptor, okay, remember that T cell can only pick out one type of epitope, but it is possible for one or more T cells to interact with the antigen presenting cell. So we have an opportunity for another T cell to access over here, and maybe they're presenting over here. We could have more than one T cell over there, as long as they have specificity for that particular type of epitope. Okay, so during this time, this is on page 361. This is the helper T cell. That's the cytotoxic T cell down there. And this helper T cell is going to start producing chemical messages, okay, that are going to not only activate the helper T cell, but also activate the cytotoxic T cell as well. And this slide just kind of looks at how there's actual binding that takes place between the antigen presenting cell and the epitope and the T cells. This is on page 362. Okay, so then here you can see these chemical messages that are going to be sent. The helper T cell sends chemical messages. It's going to actually send some chemical messages here. This is going to cause chemical messages to be sent. There's a lot of, it's very complicated. We're going to kind of skip over all of that. But then these cells start proliferating, or copying or cloning each other, and then they become specialized over here, differentiation or specialization. Okay, so we have memory T cells. Then we have some other kinds of T cells that we aren't talking about, and then memory killer T cells as well. And this is all happening in the lymphatic system. Okay, just again, another view of showing different chemical messages that are sent. I'm not going to make you remember interleukin or interferon. There's all kinds of chemical messages. Just remember that they're chemical messages. And then we'll talk about how the T cells kill here in just a minute, but then the memory cells remain and identify the pathogen again if you're exposed to it ever again. So when a cell is infected by a virus or cancer cell, a chemical message called interferon, interferons are released. Okay, and these again are chemical messages. This is just a particular type of chemical message that basically calls other cytotoxic T cells to that area. Okay. Interferon also, by the way, is like if you've ever had the flu and you feel just really run down and achy and just really tired and just feel yucky, that's due to interferon. So what the T cells do, how cytotoxic T cells destroy. Okay, they're going to bind to the antigen of the infected cell, and they're going to release a couple of enzymes called perforin, which forms a hole in the target or infected cell. And there's also another chemical or enzyme that is released called granzyme. And that chemical is going to go into the infected cell through the hole that perforin made, and it's going to induce apoptosis. Do you guys remember what that is? It's what we call programmed cell death. It basically is a series of chemical reactions that cause that cell to self-destruct. Okay, so if a cell is infected with the virus or perhaps a cancerous cell, the T cell will come over, it will bind to the antigen that's on the outside of that cell, and it will release perforin, which helps it gain access, and then granzyme will go in, and cause that cell to break down. Okay, so here they're binding, perforin makes a hole, here's another hole, granzyme goes in and basically causes the cell to break down. This is on page 363 if you want to check that out. Okay, we're going to skip that. This stage looks at B cells. So this one talked about sometimes B cells need helper T cells, sometimes they don't. We're not going to get into that too much. Most of the time, you need to have a helper T cell. Most of the time, a B cell will need to have a helper T cell. Okay, so these are going to be, remember, for extracellular pathogens. So here, let's see. I think we're going to skip this part down here. Just remember, most of the time we need to have helper T cells, sometimes we don't. Okay, we're going to kind of skip stage 2, but stage 3 of the activated B cell is going to, they're going to undergo proliferation. They're going to become effector, which are plasma B cells, and memory cells. And let's see, the plasma B cells, there we go, they're going to reside in the lymphatic tissue just like the T cells. Okay, so if they bind to the antigen, they're going to proliferate and differentiate. Let's see. And then those plasma B cells, their job is to secrete proteins called antibodies. Okay, so antibodies are proteins made by plasma B cells. They're also called immunoglobulins, or abbreviated IG. Antibodies are going to be the weapons. Okay, so, antibodies are going to kill, they destroy, or neutralize a target by activating complement. Remember that from last time? We talked about it briefly. Neutralize the antigen or promote phagocytosis. So know how antibodies work, which is this bullet point here. and also know that antibodies are proteins. So a plasma B cell is going to produce, so this might be a good slide to star. It is on 367. Oops, 367. Here's our plasma B cell. It's going to produce antibodies. And the antibodies are basically like B cell receptors, only they're free. They're floating around, and they will attach to antigens. Okay, so pretend this particular antigen is a toxin. Okay, these different antibodies, there's one here, there's one here, and one here, they will attach to that toxin or that antigen and form a complex. And now remember, even if it's a toxin or a bacterium, if it can't attach to a host cell, it can't cause any problems. So it basically blocks or prevents binding to the host cell. Complement, that's part of the innate immune system, remember from last time. And there's a lot of different ways that complement works, but they can burst the cell, cause opsonization, which is coating. Okay, where the antibodies can coat this bacterium here and the phagocyte can identify it better, or stimulate inflammation. Okay, so they can neutralize, activate complement, or cause an increase in phagocytosis. Okay, precipitation is basically where things just kind of settle out. Agglutination is clumping. If you guys have done the blood typing lab with me before, you remember the clumps that form if you add the serum with your blood type and it's a match, they'll form a clump. Or again, opsonization, where you coat the outside. All of these things draw the phagocyte's attention to the pathogen, and the phagocyte can then eliminate it. So definitely know how antibodies can work. Okay, this slide here. So we talked about that. Antibodies increase phagocytosis. We've all covered all of those. Precipitation, agglutination, and opsonization. Which is the coating process. Okay, so this might be a place where you stop and review for a little bit too. Okay, so here's a close-up view or drawing, I guess, of an antibody. Again, it has two binding sites here and here, and it will only bind to a specific shape. It's kind of like an enzyme. Remember, enzymes could only bind to substrates with specific shapes. Same thing with antibodies. It's not going to bind to this particular epitope on that antigen, only the correct one. I'm going to skip this. And here there are 5 different types of antibodies, and you're going to need to know this as well. It's on 368. You'll need to know the 5 types of antibodies, and the different types of antibodies are called isotypes. Okay, so the first one is IgG, and this is the most abundant. Most of the antibodies circulating around in the blood are going to be IgG. It crosses the placenta, so it can help baby, okay, if mom is pregnant. The others do not cross the placenta. And it has a half-life, or basically a time before half of them are destroyed is 21 days. So they're going to be the longest lasting antibody in the body. IgA is the next one here. It has a half-life of only 6 days, but it is secreted in milk and other body secretions. Okay. IgM is going to be the largest in size, and it's also going to be the first responder. IgM is usually the first antibody produced. Okay. What else was I going to say about it? I think that's it. IgE is used to fight parasites and is going to be at high levels in allergic responses, or if you have allergies. And then IgD, not really much known about that. Okay, so those are the 5 antibodies. Again, IgG is the longest lasting, has a lifespan or half-life of 21 days. Crosses the placenta, we know that. IgA has a lifespan or half-life of about 6 days, and it is found in body secretions. IgM, okay, again, let's see. Made early, it's going to be the first responder. And it's going to be the largest in size. IgE fights parasites like worms. And they also help release histamine, leukotrienes. They're involved in allergic reactions as well. And then IgD, not a lot really known about that. Okay, if you like memory devices, this might be a good way for you guys to remember some of the different agents that are involved with adaptive immunity. So if you're interested, this one's kind of silly, you can kind of read through that, and that might help you as you study and try to remember what some of these cells do. One thing I did forget to mention, I just noticed this, is that plasma B cells, which is this guy here, they produce about 2,000 antibodies per second. And they are, ammunition producing machines. They are crazy busy when they are producing antibodies, but they don't live very long because they wear out really fast. But I just thought that was interesting. They can produce 2,000 antibodies per second. And those are proteins that they're making. So they're undergoing transcription and translation. Okay, it's all tied together. Okay, so once the threat subsides, once the immune system's gotten the threat pretty much eliminated or pretty close, effector cells start to die off, memory cells stay, and the memory cells stay and provide immunological memory, and that memory is going to allow for very fast reactivation of the adaptive immunity if you're exposed again later, So remember, our primary immune response, the first exposure, takes at least a week for T cells, B cells, antibodies to get to a higher level. Secondary immune response, the secondary exposure is coordinated by the activity of the memory T and B cells. So secondary exposure is going to lead to the secondary immune response. And in the primary immune response, remember IgM, they're the first responders, They're not as tough and they're not as effective. They're good. We like them. We're proud of them. We're glad we have them, but they're not quite as effective as IgG. So when you have a secondary immune response, IgG comes out a lot faster and a lot stronger than it did in the first response. I'll show you a slide of that here in just a second. So second response to the same antigen works really quickly and generates a higher titer. And a titer is basically just the concentration of an antibody in a substance like the blood. So if you have high titers of antibodies, that means you have a lot of antibodies per unit of blood. So here's first exposure, first response, second response, and these are on the same scale. Both of these graphs are on the same scale. You see IgM, takes a few days to get going. IgG's out here after 2 weeks. Secondary exposure, look at IgM. Look how high IgM is just after a couple of days. So again, this is kind of giving us the premise and why we vaccinate, is to get that secondary immune response. And sometimes, and we'll talk about this when we get to vaccinations, but sometimes you have to give boosters, Tetanus is every 10 years, because your memory starts to wane you give that booster and it just gives it a little boost. Okay, so immunological memory comes from a couple of different ways. Okay, so we have active immunity and we have passive immunity. We have naturally acquired and we have artificially acquired, and we can mix and match those terms. Let me move this out of the way. So one of the things you'll have to do on the test is determine if this process is naturally acquired passive or artificially, you'll have to figure out which one this is. Okay, so here's how you can do that. So, is the process natural, or did it come from a healthcare intervention? Was it given to you by a shot or an injection or an IV or something like that? Was it an artificial process or was it a natural process? Okay, so you look to determine if it's artificial or natural. You determine then who made the antibodies. Okay, if a person made the antibodies themselves, it's active. They made the antibodies. Your own body made the antibodies, it's going to be active. If someone else made the antibodies, like in this picture, mom made the antibodies and transferred the IgG across the placenta. Okay, so in this case, it's a naturally acquired process because it's not coming from a medical intervention. So naturally acquired, and it's passive when we're considering the baby because mom made the antibodies, not baby. Okay, let's look at another passive one. Actually, let's not. Let's look at this other natural one first. So a person getting sick, okay? Say you get the flu, okay? You naturally got it, okay? You yourself made the antibodies. So it's a natural process and your immune system was active, okay? Think of it that way. If your immune system was active in making the antibodies, it is going to be an active process, okay? So baby wasn't active. Somebody else made the antibodies. So that's a passive process, okay? If you need to stop there and think that through, go for it, because you'll definitely need to know the differences between these, okay? So natural process, if the person did make the immune system, it's naturally acquired active, or make the antibodies. If the person did not make the antibodies but got it naturally, it would be naturally acquired passive immunity. Over here, we have an artificial process. Okay, so we got injections. So here, a person is getting a vaccination. Okay, that's a medical procedure, so it's going to be artificially acquired. But when you get an injection from through a vaccine through an injection, your body is making those antibodies. Your body is building up the memory, so it's an active process. Okay, so artificially acquired, active. Then say a patient gets bit by a rattlesnake and they go to the ER and they get an antivenom or some sort of anti-serum. Okay, it's a medical intervention, so it's going to be artificially acquired, but it's passive because usually a horse or another organism has produced those antibodies. Okay, so let me go walk you through these slides here. So naturally acquired, active. Okay, you've contracted the infection, the memory cells and antibodies are formed by the patient, and it's going to confer long-term immunity. Okay, maybe just a few months, or it could be years and years and years, or a lifetime. Just depends on the situation. Okay. Artificially acquired active, this would be the vaccine. Okay, because it's active, it's going to give long-term immunity. Naturally acquired passive, where you get antibodies from someone else, okay, like mom. There's going to be no memory cells or antibodies. Okay, because remember the half-life of those antibodies, the longest one was what, 21 days? Okay, so after those antibodies have all been broken down by the body, you have no protection. Okay, so once mom, say mom is nursing the baby and she's providing antibodies through the milk, once those antibodies break down, there's no longer protection. Okay, temporarily protects the baby. Say mom gets the flu, she starts producing antibodies and passes those on to the milk. That will be protective to the baby, but it's not going to protect the baby 6 months from now after it stops nursing. Okay, and then artificially acquired passive, that was where the patient receives protective antibodies from a medical treatment, usually a horse. They would get what's called an anti-serum that's going to help protect them from a toxin or a venom. It provides temporary protection, okay, no lasting protection. Okay, and a patient, you know, if you get bit by a rattlesnake a second time, 6 months, a year later, you'll still have to go back because your body didn't build up the immunity. Now, that being said, there's people that are snake handlers that get bit dozens of times over the years. They will eventually build up immunity, but just your regular old person getting an anti-serum is not going to build up memory. And also, you can have what's called an serum sickness, where your body actually attacks the serum and has an immune response to the serum that's trying to protect you. So there is that possible scenario there. Okay, so that finishes up chapter 12. There will be a mastering homework that's going to be due, and I'm going to look for some videos, and I will post those on D2L for you guys to watch, just to kind of enhance your learning on this, because I know immune system's really tough, and it's a tough concept. So I'm going to try to find some videos, and I will let you know on the daily checklist if any of those are going to be required. Okay, so again, let me know if you have any questions about this material. We can try to follow up and then be looking for some of those videos. All righty, we'll talk to you later.